For the most part, especially in the confirmatory stage, trials are like really not terribly different than the way, Jonas Salk did his polio trial. And it's hugely ironic to me that as biostatisticians, sort of, my job is to identify whether the newest, latest, greatest things work, like lasers and robotic surgeries and, fascinating billion-dollar compounds. And yet we're still using the same tools to evaluate whether these things work that, we invented in the 30s to test crops and fields, that we're not doing lots of new things statistically. And there are lots of reasons for that, but it's worth stepping back to say, can we do something more interesting, more efficient, that treats patients better and that gets our answers faster? So to try to illustrate that, I'm going to walk through one particular trial here and maybe show how this works. So the question in this trial is how to treat patients with static epilepticus. So these are patients who are seizing. Basically the ambulance comes and they don't stop seizing after being treated with benzodiazepines. And there are 3 drugs that are commonly used once they get to the emergency room: fosphenytoin, levetiracetam, and valproic acid. And it's not really clear which of these 3 things is the best. There's no guidance that says which of these 3 things is optimal. And I guess different emergency room docs use different ones of these. So the idea for this trial, which will be in NIDK or NINDS funded, is to figure out and provide information to clinicians as which of these 3 drugs is the best. So, much debate about endpoints, and the endpoint here was, well, you want the seizure to stop quickly, you want the seizure to stay stopped, and you don't want the drug that does this to have any other bad effects. So it was this composite endpoint, and this is a case where we learn quickly. Like when you do a cancer trial, ideally the patient survives, but even if the patient dies, that may not happen for months and months. So it's a long time till you get the endpoint. Here we know the endpoint very rapidly, and therefore we can adapt to that and learn as we go and update the design process. So there's no control group here, right? It's not like a regulatory trial where we're trying to prove something's better than a placebo or trying to prove something is non-inferior to a standard of care. That all these 3 drugs start out equal. All these things are available. Only one of these is actually approved with a label for this indication, but the others are
5 other generic.
Of course, well, what drug's the best? How sure are we of that? Is one of these 3 drugs worse than the other 2, and how sure are we of that? So let's for a minute try to forget what we know about clinical trials and think about sort of this comparative effectiveness trial that we've all done, right? We've probably all moved to a new city and started a new job. So I live in Orlando and up by A, so that's College Park, and the new College of Medicine in Orlando is down in the bottom left where you see B. So I could get to work by kind of going down and over on the highway, over and down on the highway, or taking surface roads through, right? So none of us, absolutely none of us, would design a clinical trial, right? Or seek out a statistician and say, hey, I need to figure out how to get to work. How should I do this? And the statistician says, well, what's clinically relevant, right? And you say, well, if I can save 5 minutes, that's the way I'll go. But if I can just save 4 minutes on average, I'm just going to go like the way without tolls. And you wouldn't design a trial and he wouldn't give you randomization envelopes that every morning when you make your coffee at home, you rip open a randomization envelope and then go that day. And then 6 months, and when I powered this trial, you would have to go each way 40 times, right? And so that's 40 times is 120 days, which is about 6 months of work days. And then the statistician would tell you, do not calculate an average as you go. Don't do that, right? 120 days from now, I will do an analysis and then tell you how to go every time thereafter. That's not how any of us make decisions, right? Like we probably talk to someone around school and figure out who lives up near me and then go talk to that person and say, well, how do you go? And try that and maybe try another way. And so you figure out, well, the first way here is actually the fastest, but maybe someday you don't have morning meetings and it's not a super busy day and you try one of the other ways. And then you go, oh, now I know why I never go this way. Or you go, oh, that wasn't that bad. And you try it again a little more often, right? That's the way we make decisions. And for the most part, we get to work an optimal way. But that's not the way we do clinical trials. And well, why not, right? In part, it's not pre-specified, and clinical trials should be pre-specified. Research needs to be reproducible. That's not reproducible. But we can formalize that in a way so that, for the most part, we end up going an optimal way, but we still try other ways. But we can do that in a predefined setting. And plus, being in a predefined setting allows us to know error rates. Like right in this process, who knows what our error rates are? But we can design it in such a process where we can actually identify.
So the USP right there, the official compendia, right? This is sort of product quality standard. I'm just going to walk through quickly one example trial. So this is just one simulated trial here. And you can see the success rates for the 3 drugs, the probability that each of these is the maximally effective drug. And I don't get into sort of the Bayesian math here, but that's straightforward.
But you can sort of see VPA had 64% versus 55 versus 51, and there's an 88% chance VPA is the best drug, best defined on this composite endpoint, versus the other two had a 9% and just a 2.5% chance of being the best in terms of this composite endpoint. And then those translate into allocation probability, so we're putting 2/3 of the next 100 patients on VPA, 22% on fosphenytoin, and just 12% on levetiracetam. So when we do that, we have our 100 new patients, and you can see that the success rate in this next wave was actually higher on fosphenytoin. So instead of VPA being 9% better, it's now just 5% better. So we're still putting, not many patients on levetiracetam, and the majority of patients still on VPA, but not quite as many. Instead of 66% in the next round, it's just 57%. So we're still putting more patients on the best drug, but more patients on fosphenytoin. And we keep updating this process. And so in our next 100 patients, VPA did the best again. So we had a 68% response rate there versus 53 for fosphenytoin. LVT is still the worst. We almost get to the point here. So there's an 8% randomization probability to LVT. If that were 5%, we would have just zeroed that out for the next round. But there's still a few here. And this is one of those questions where we show simulations during the design process to the epileptologist and say, well, there's a very low chance that this is the best. But we're still randomizing 8% of patients to that. Do you still want to do that, or should we zero this out sooner? And they say, well, no, lots and lots of docs use this. There's a literature on this. It's okay that we're still putting a lower rate there. And of course, if this were a fixed trial, we'd still be putting 33% here. So we're still doing something better than sort of what might be conventional. And of course, a DMC is observing this too. And if there were other factors coming up, the DMC always has the prerogative to recommend an arm be dropped. And then this last wave of 100 patients, we see, VPA is still doing better and better here. And by the end of the trial, I didn't say that the maximum trial size here was 800, but at 600, when we see response rates of 52, 58, and 69, there's a 99.2% chance here that VPA is the best drug. That's higher than the 97.5% time where we said we'd stop the trial. So we stopped this trial and identify VPA as the best. So the results that would get reported would look something like this. So these are Bayesian posterior probability distributions. And we can see that VPA, which is the best drug, has the tightest precision. So that's good, right? I mean, if everyone is now going to shift towards VPA, or that's going to be the recommendation, it's nice that it has the best precision of these three drugs. And that also meant that the majority, or almost the majority, the plurality, but almost half the patients in the trial actually got the best medicine, instead of just 1/3 of the patients getting the best medicine. And we can report the probability that it's the best and the probability that LVT was the worst drug here. So this is what the results of this trial might look like. But the key is, more patients got good medicine in this trial, and we got our answer sooner than we would have gotten in a fixed 800 patient trial. So let's see so that was just one example, of course, but let's see how this will work as a whole. So comparing adaptive randomization with fixed randomization. So we had the adaptive randomization process I described on the left, and fixed randomization, we can still have early stopping, but fixed just 1/3, 1/3, 1/3 randomization on the right. So in the null case, when all these drugs just work half the time, we see this is the power that we identify one of these as the best. So that's a type 1 error, right? These drugs are equal to one another. So that's a type 1 error, and our type 1 errors are low in both settings. Sample size is actually a little bit higher when we're doing adaptive randomization. But in the situation where one of these is superior, so one is 15% better, the trial was powered to identify a 15% difference if it exists. So in this case, we have 90% power with adaptive randomization. Power is a tiny bit less. So this is based on 100,000 simulated trials, like I just showed you. We did that 100,000 times. Power is a little bit higher when we have adaptive randomization. The average sample size is a little bit lower. And the key is then, instead of randomizing 1/3 of the patients to the best medicine, we randomize 47% of patients to the best medicine. So here's a trial design that has higher power, lower expected sample size, and more patients getting good medicine when one of these drugs is better than the other two. In a case where, instead of being 15% better, it's not powered for this situation, but where you're just 7.5% better than one, power is still higher in the adaptive randomization setting. Sample size, effectively the same, but a tiny bit lower. But the number of patients, or the proportion of patients who are randomized to the best drug, increases again from 33 to 47%. Where it does fail, there's always trade-offs here. Because we're randomizing away from the worst drug, if there's a setting where 2 drugs are equally good and the third one is bad, well, the fixed trial is still putting a third of patients on that bad dose, right? And so it has higher power. The lower number is the power to identify the worst drug. So this still has 80% power to identify that worst drug. Here, that drops to 69% because we're putting fewer and fewer patients on the bad medicine. So, if you're a patient in this trial and there's mortality associated with not stopping seizing, that's a trade-off, that it's bad to identify it's the worst, but, we're putting fewer patients on what is bad medicine. So the summary here, this is a comparative effectiveness setting, but in particular, comparative effectiveness is a really great setting for Bayesian adaptive trials, that we can get higher power, smaller sample size, and treating more patients with the best therapy. But these same things apply in, particularly phase 2 trials, where you're looking at a whole bunch of doses and identifying what's the best, where we need to identify, the best dose where we're getting good efficacy, but not bad adverse event rates yet. Instead of just picking 3 doses or having to ramp up, we can do something adaptive and look at a much broader dose range, or especially when we're talking about combination therapies. We can look at a grid, and you obviously can't do a fixed trial with 3 doses of drug A and 3 doses of drug B, or it's hard to look over that whole range. But with adaptive randomization, you can have many, many combinations and sort of hone in on the best area of action. So the idea is, we want to do interim analyses early and often. Here, we did it every 100 patients, which is actually not very often. But this was an emergency medicine trial. These patients aren't giving informed consent. It's not like a cancer trial where you give informed consent. You can put information into a computer, get a randomization probability. Here, basically the nurses or the doctors just grab the bag that's on top and give it to them via IV, because these patients are in the emergency room and seizing. And every time we update randomization probabilities, we actually have to call the hospital and have them reorder bags. And of course that involves human error, so we figured the less we did that, the less chance for human error. But if you're doing something like a web-based randomization, we've updated probabilities every week and even every day in some circumstances. But we steer patients to better therapies. We stop the trial as soon as we know the answer, which is the key, because the idea is, we want to do things efficiently. If we want to lower the time from drug development from 9 years to 5 years, the faster we can do trials and the faster we can report answers and get to the next stage of development, the better. And it's really just sort of like the way we get to work, a natural way of sort of learning and making decisions. None of us blind ourselves to knowledge and then, make a decision 3 years later. We learn as we go, we adapt, so we can make decisions as efficiently as possible. Thanks.